So, this was the slide we left off on Friday afternoon, the refractory period. Before we start diving into the new material, let's just review briefly so we can get our frame of reference. During this presentation, we were talking about action potentials. Action potentials are a wave of depolarization or some electric current traveling across the surface of the cell, particularly the axon of the neuron in this context of the nervous system. And as we look at an action potential, we need to have sodium voltage gated channels open up at threshold, sodium ions rush in. At that positive 35 millivolts, the sodium voltage gated channels are closed or deactivated, and the flux of sodium ions stops. Potassium voltage gated channels open at positive 35. Potassium ions start rushing out, and then we hyperpolarize the cell, and when the voltage bottoms out in the middle of the hyperpolarized state, potassium voltage gated channels close, potassium ions stop moving out, and then to get the neuron, to get the axon of the neuron back up to resting membrane potential with the correct concentrations of potassium and sodium on the inside and outside, we have the sodium-potassium pump constantly chugging along, resetting the ion concentrations. Now, during this action potential, there are some periods of time where it's absolutely impossible to have another signal sent, and another period of time where it's really difficult to have another signal sent. The period of time where it's absolutely impossible to have a second action potential sent is the absolute refractory period. And when we look at this absolute refractory period, nothing can initiate another action potential during this time period. And this absolute refractory period is going to last as long as sodium voltage gated channels are open or as long as they are inactivated. We didn't spend a lot of time going into the activation, deactivation, opening, and closing of sodium voltage gated channels, but it's worth marking on your figure that typically at this point during the peak voltage, sodium voltage gated channels are deactivated, and then down here, sodium voltage gated channels are closed and reactivated during that period of time. So during this period of time, during the absolute refractory period, there's some practical implications. This gives us a theoretical maximum frequency to action potentials. It means that we have a max rate that we can send action potentials down the neuron at. It also means that there's some mandatory rest time. Physiologically, we can only send the signal so quickly. We must have time to rest and reset the axon during this absolute refractory period. And then we have the relative refractory period. During this relative refractory period, it's still really hard to send another signal down the axon and really hard to send another action potential down it. But we can do it if we really, really try hard. So to overcome this the relative refractory period, we need to take the stimulus signal, we need to take the membrane voltage during this hyperpolarized state and bring the voltage all the way back up to the threshold. And bringing the voltage all the way up to threshold when the membrane is hyperpolarized means that electrically we need a stronger signal. When the membrane is at the resting membrane potential, when we're at negative 70 millivolts, we only need a 15 millivolt stimulus to get up to threshold. But when the membrane is hyperpolarized, you need a 20 millivolt, a 25 millivolt, a 30 millivolt stimulus because the membrane is even more negative and you have to overcome that increased negative electric potential of the membrane in order to initiate a signal. Practically speaking, during the relative refractory period, we will only be able to feel the really strong signals, but weak signals we tend to filter out and zone out. It's one of the ways that we can accommodate to overstimulation. These absolute and relative refractory periods help us also to filter out repetitive stimuli, but to pay attention to the really strong stimuli or pay attention to changes in stimuli strength. When we look at the conduction of a signal with our axons, There's myelinated and unmyelinated axons. And these different kinds of axons, the myelinated and unmyelinated axons, are going to send signals at different speeds. When we look at an unmyelinated axon, it doesn't have oligodendrocytes or Schwann cells to myelinate it. Instead, it's one naked axon. And that entire naked axon is going to have a lot more sodium and potassium voltage-gated channels that need to open and close. And because there's more ion channels that need to open and close throughout the length of this axon, and there's more steps involved, the overall speed of the signal is going to be slowed down. So when you think unmyelinated axons, I want you to think slower action potential speeds. This process of having this unmyelinated axon is going to give us a kind of action potential conduction called continuous conduction. Our action potential will always head one direction down the membrane. And as we're looking at this action potential, remember, the action potential graph, this graph that's up on the screen, shows the changes in voltage at one specific location on the axon. So as we look at our axon, here in yellow we have a refractory period of time. Here in red we have an action potential in progress. And then here in green we have a membrane that's at the resting membrane potential. And as our action potential is progressing, because of this refractory period, the absolute refractory period on the downstream end, this keeps our action potential progressing one direction down the cell. In many ways, it is exactly like dominoes falling. So if you ever think of dominoes falling on the floor, you line up your dominoes, you tip one end, and they all fall the other direction. They typically will never fall backwards because the dominoes have already fallen over, and you can't have a domino fall over a second time when it's already been knocked over. Let's talk about myelinated fibers. In the central nervous system, we have oligodendrocytes myelinating our axons. In the peripheral nervous system, we have Schwann cells myelinating the axons. In both situations, we're going to have little gaps in between myelination sites. The traditional name for this is the node of Ranvier, and this node of Ranvier can be visually seen right here as the space in between myelinization points. This figure up on the screen overemphasizes the width of the node of Ranvier relative to the size of the Schwann cells. Think back to lab when you were looking at nodes of Ranvier last week during lab. Those nodes of Ranvier are almost indistinguishable at 400 times total magnification. Primarily what most individuals, myself included, look for is the cleavage furrow in between two adjacent Schwann cells. Now, when we look at this signal conduction, if we have a myelinated axon, we're going to have sodium entering the cell at the myelinization point. And then the voltage will travel through the axon to the next node of Ranvier. This process of skipping sodium voltage-gated channels and potassium voltage-gated channels is ultimately going to speed up the conduction speed of the action potential. So instead of having continuous conduction with more ion voltage-gated channels that we have to open and close, this myelinated conduction, sometimes referred to as saltatory conduction, is going to be a much faster way of sending the signal. A common misconception that students have is that the signal will jump from one node of Ranvier outside of the cell, outside of the axon, to the other node of Ranvier. That is a slight misconception. The signal instead jumps within the axon to the next node, and then from that next node through the axon to the adjacent node. So that's a better representation of the jumping that occurs. For those of you who have spent any amount of time studying physics, these myelination sites are they're slightly analogous to capacitors and charge buildup on opposite sides of capacitors. But that's not a perfect analogy. So as we're looking at myelinated axons, they send the signal faster than the unmyelinated axons. And make sure you write in the margins that this is also known as saltatory conduction. While we're talking about axon speeds of action potentials, it's also worth writing in your margin notes that large diameter axons send the signal faster, narrow diameter axons send the signal slower. So by alternating the myelinization and the size, we have two different ways of manipulating how quickly the signal is sent on our axons. And I believe that's all for this presentation. Let's jump into our next one. Synapses and neurotransmitters. Question. Saltatory conduction is the myelinated conduction.